Okay, afternoon. Welcome in the next lecture from biology. Today we are going to focus on transports via plasma membrane. I remember that I promised to you last time that we are going to provide some calculations. However, as I check it, the presentation is a little bit longer than I expected. So the calculations will be next time. So please keep in mind that next time you will need a calculator. So today I will just I will just give you some background from the theory, from transport via plasma membrane, and in the end of this lecture I have prepared for you some mini test. So there is a big chance to receive some extra points for your credits. That's, I think, good information. So let's start. Yeah, presentation is recorded. We'll share the presentation. Okay. So today, membrane peptides and transport agents. In general, we are going to talk about the transport via plasma membrane using facilitated diffusion and using active transport. So these three we are going to mention. So first of all, let's introduce a little bit the physical types of transport via plasma membrane. So just first of all, just make a scheme in your mind. So you have a barrier, which is a membrane. So it could be in your mind, it could be a wall with doors or something like that. So what is possible? From the physical point of view, we can transport just one particle from one side to the another side. Maybe I can draw it. Okay, uniport. So we have a wall. I can draw it like this. So one particle goes from one side to another side. And the same particle just via this barrier. Symport, it means that two different particles, the red one and blue one. Okay, blue one. It's not easy to draw with fingers. And this active one and the red one. Okay, they are going together. So it means that they are supported somehow. So the blue one supports the transport of the red one, for example, or the blue one supports the another transport. So this is the second case. I think you can imagine quite easily. And antiport means that in the same point, in the same time, we have a blue particle which goes in one direction, and we have a red particle which goes in the same moment to the opposite side. Again, this antiport supports somehow the transport of both particles. So this is the physical point of view. The chemical point of view, which is maybe more familiar to you, as the passive, facilitated, and active diffusion. For passive diffusion, we are using only chemical gradient. So chemical gradient. Okay, gradient. Chemical gradient of the concentration. So it means that we have a wall, and here are many particles, like this, and on the other side there is practically nothing, maybe zero, known particles. So naturally what happens if I open the doors, which can be in the membrane, in a wall? So what can happen? The particles from side A to side B will smoothly diffuse through the pores, through the doors. Maybe you can imagine this process. Just we have one big room, and in this room there is a wall. And one half of this room, maybe I can draw it also. So we have such a big room. There is a wall, and people are only in a half of this room. In the second one, there is nobody. So just call it A and B. And if we remove this wall or open the door in the wall, naturally people will spread to the second room, and they will be in some time. So also it's the description in time. So this is in time number 0, t is 0, and in t maybe in 10 minutes. So number 2, okay, 1 second, or 10 minutes, maybe 10 minutes. People will be spread in whole room constantly. So is it clear, this approach about the passive diffusion and what is the concentration gradient?
Yes.
Concentration gradient. So what is the difference between passive diffusion and facilitated diffusion? It's the principle is quite similar, because for passive diffusion I am using, or for the process is used the concentration gradient. For the facilitated is also used the concentration gradient. However, the facilitated diffusion or transport is characterized by some agent. It could be a porine, it could be a transporter within the plasma membrane, which allows to transport some chemical, some particle from part A to part B, from outside to inside the cell, for example. Normally, this facilitated diffusion is used, or facilitated transport is used according to the concentration gradient. This is the first point. And also it's used for the particles which are maybe badly dissolved in plasma membrane. They have too much interactions with plasma membrane, so therefore it cannot be smoothly transported because of interaction with plasma membrane. So for this purpose, we have a particle, some ion, which can be covered by something, and in this particle is transported. So that therefore we call it facilitation. Is it clear? Just say yes or no.
Yes.
Okay. But in contrast to this transport, the active transport, it's usually against the concentration gradient. So it means that I have a room. In the room, it's just the first. Wait. I have the first case, like this one. Maybe I use different color. Okay, this one. Like this one. I have a room. In the middle of room there is a wall, and all the people are in part A. However, I would like to transport into part A more people, so I am pushing them into the part A, and on the other hand, the part B is completely empty, or more or less empty. So I am pushing people against their concentration gradient. So it's the same like with particles. They are not really willing to go to crowded room, so they will be better for them. It's natural just to spread in a big room, in an empty space. However, sometimes I need to transport some particles, also people, to some crowded space. So that's the way that I need some energy. This is the point. So for active transport, I need some energy, and also the transport goes against the concentration gradient. So in this opposite way, like drawing this with purple color. Is it clear?
Yes.
Okay. So you will check it by the test, sure. Okay. Okay, could you hear me? No, so maybe it was my problem on my side because of the weak internet connection, but now it's fine. Okay, let's go back. About diffusion. Normally, you can clearly imagine that people, also animals, everyone is moving. Yeah, movement, it's a typical characteristic of the majority of living organisms. However, is it the same also for particles? Yes, it is. Molecules, atoms, all the chemical agents, they are moving. And the movement, it's random. There is really no sense that they will force their moving, so therefore they are moving along their concentration gradient. This is the first point. And also they also provide some random walk, so it means that the particles are just making a long way, not a direct way, and by this way the solution, the air, it's mixing. Maybe it's better to show you the video, so we will see how it works normally.
Vám teď ukážu dva nadpisy, což je zlatý grál, text vteřiny. A jeden z nich vydělává.
Powered by thermal energy, molecules are constantly in motion, called Brownian motion, which allows them to diffuse through cells in a random walk. Our simulation shows the degree to which a small sugar molecule on the left and a larger protein on the right explore the interior space of a cell, here shown as a cube with a 10 micrometer side. The animation represents 1 second in real time. Note that the paths of the molecules in the second simulation are different from those in the first, thus showing the randomness of Brownian motion.
Okay, so is it clear from the picture, from the video? So random Brownian motion or movement or random walk, it means that all particles, atoms, molecules, they are moving, they are rotating, they are shaking, also they are so-called walking. Yeah. So however, this walk is stochastic, it has no direction in the moment. We can just observe from time 0 to time maybe 10, 20, just to some determined value, and by this way just measure the distance that particle, one particle get from one space to another space, from the point A to the point B, and we can measure the distance of the walk. However, between these 2 points, A and B, there is a pretty much longer way. So it means maybe if you are go outside with your child, little child, and you are walking, yeah, you have clearly direction from A to B, from your house to the shop, and you walk just direct. However, your child is running around of you, so it maybe it's quite similar as the particle. Just always keep in mind that the driving force of the transport of every motion and diffusion is concentration gradient. Also the quality of diffusion, so it means how easily can get one particle from one point, from point A to point B, is characterized by the diffusion coefficient. So it means that small particles can be faster. They can make more movements in general. Bigger particles or particles with just complicated shape will provide the movements more difficulty in the movement because of the interaction. You know, I will show you. So, for example, I have 2 molecules. This one and this one. This is a ball. Could you see it? If I'm showing just a little picture downstairs or upstairs?
Maybe if you move it a bit up.
Okay. We'll just. Okay, maybe this is better. Okay, this is better. I have two particles, two molecules, yeah? This one is a ball, this is a cube. It really depends on also the quality of the ambient. So in some ambient where the ball can go smoothly, this particle will be faster, the diffusion will be faster, and also it could be characterized by the diffusion coefficient for balls. And in the second case, we can also characterize a diffusion coefficient of cubes. Naturally, molecules are not only balls and cubes, we have different shapes of them, and they also have a charge. It also really depends if the particle or the molecule is moving in the field which is created by negative charges or positive charges, because the negative charge will really, so the positive and negative charge, it attracts together. So if I have a negatively charged field, for example, and the particle which goes through this negative field will have strong interaction with the negative charge of the field. So therefore it will be captured inside and a little bit struggled, and the movement couldn't be possible. So this point is clear?
Yes.
Yes.
Okay. Great. So, what influenced the diffusion coefficient? Maybe again, I will just make a little compression of all information. So the shape and the size of the molecule, and also the charge of the molecule, if it has a positive one, neutral, or negative one. Also, we can talk about the diffusion of gases, we can talk about the diffusion of proteins, about sugars, all the substances. So I think this is too special maybe. First of all, I would like to talk about some general movements, and in the end of this lecture, I will give you some the most important examples. Now I would like to explore a little bit osmosis. This is very important because the cells in solvents or solutions with different concentrations, they can be swells or shrinked. So it really depends on the concentration of salts inside the cell and outside the cell. So first example, if we have a solution which is called hypotonic, what it means. So
less than body temperature?
Yes. Great. So we have a cell. Cell it's really determined by the evolution, the concentration of salts inside the cell. Yeah, it's a salty bag actually. And this is first point. Yeah, in cells I have some defined concentration of salts, of ions in general. And also because I would like to, or the cell, for the cell life, it's very essential to keep this concentration constant usually. That's the reason that the plasma membrane, which really surrounds the cell and defines the shape and inner space of the cell, is not permeable normally for the majority of ions which are inside. So it means that the ions or salts, I can talk about salts, maybe you are more familiar with salts, but it's ions, in fact, they cannot go inside the cell and they cannot go outside the cell. That's it. So it means that the membrane is permeable only for water. Is it clear? Just I will draw it maybe to be sure that it's really clear. So this is a cell, just a cell. Inside, so inside is water, outside is water. Inside are some salt or ion particles. Across the membrane can go only water. So water can go inside and outside. The doors for ions are closed. Is it clear? Could you accept this approach?
Yes.
Okay, so just mentally think about it and go with me. So we have a cell with some concentration inside the cell. The value of the concentration is not essential to just know right now. However, I immerse this cell inside the solution which has 0 or very low concentration of salts, ions, much lower than is inside the cell. What happens if you know that the plasma membrane of the cell is permeable only for water molecules? Marek.
Could you repeat the question?
Yes,
like,
I can.
Yeah, I think I understand, but I'm not sure.
Okay, I have a cell.
Mm-hmm.
In the cell, there is some defined high concentration of salts, of some particles. Yeah? I immerse the cell into the pure water. There is no salt anymore outside, yeah? Or just a little amount. Much lower concentration than inside the cell. And the cell is surrounded with plasma membrane, which is permeable for water only. Permeable means that water can go across the membrane, nothing else. What
So
happens?
I would imagine the cell to probably suck in some more water, but the ions would stay inside.
Yes, because of the concentration gradient. That's good. Yeah, thank you very much. Because water will try to dilute the concentration inside the cell. So we can see that the cells are just bigger and bigger, and maybe they can explore, yeah, because of the sucking of water from outside ambient. Eva, is it clear?
Yes, it is.
Okay, I will ask you namely because I wait too long for some reactions of you. Eva, stay with me. A little bit different example. What happens if I have the same cell and I immerse the cell to the solution which is very concentrated salt solution? So the concentration of salts is higher in the solution than in the cell. What happens? Only water can go through.
Well, the water will go out of the cell into the surrounding liquid that has higher concentration.
Yes, very good. And why?
Because of osmosis, because the concentration gradient wants to stay the same.
Yes, yes. Excellent. Thank you, Eva. And Sunny, are you there?
Yes, I'm here.
Okay. Third questions for Sunny. What happens if I have a cell which has some defined concentration of salts inside? I immerse this cell into the solution which has exactly the same concentration of salts inside. What happens?
It's not nothing can happen because
Nothing. That's great. Okay, excellent. So you understood pretty well. So now we just follow 3 extreme cases: hypotonic solution, hypertonic solution, and isotonic solution. Yeah. So in this picture you have everything. Great. Now we are going to move to facilitated transport. It's very important to keep in mind that still, if it is facilitated, it goes along the concentration gradient, not again, not contra. Yeah, it goes along with the concentration gradient. So this is very important just to remember. Still, force is the same one, concentration gradient. However, naturally, because of the chemical quality of the substance which can be diffused, it requires some assistance, some help. Yeah? We have 3 types of this facilitated transport. The first one, we have some mobile carrier. The second one is that the particle goes through a special pore or channel, or it's transported via intermembrane protein. So these three types are possible. I will show you some pictures. I have them, but first of all, just to talk briefly about every transporter separately. What can we say about the mobile carrier? It's, from the chemical point of view, it's usually a protein or uniforous antibiotic. In the first case, if it is a protein, it's usually a transport protein or translocase or permease. So transport protein, so something. So we have a membrane. I will draw it again. We have a membrane, and in the membrane there are some proteins. The proteins can they can transport the chemicals from one side to another side, and the way how they are transport them, it's called by the translocase. So it means that the protein is in one side, just became in contact, the particle gets inside, and together with all the complex go outside, and the particle is released from the transporter. Permease, so this is translocase. Permease is that the molecule goes through and helps the particle goes along the plasma membrane. Yeah, it permeates, like it makes a little pore, just open it and help to the particle go through. Also, in chemical engineering, is used some uniforous antibiotic. Usually they are soluble in lipids. So as I mentioned last time, so the inner part of the plasma membrane, it's created by fatty acids. So it's a lipid, so it has an innate interaction with this lipid part. An example is valinomycin or grametidine A. So I will show you them in picture. So this is valinomycin. It's like a shell. So it's like a maybe like a circle, and this circle has the inner space which is empty, and the particle which can be transported just is hold on in this circle and goes through the membrane. Yeah? It's like a wrap for the potassium in this case, and it's quite easy. So valinomycin really helps. Yeah, it's antibiotics. Valinomycin really transport potassium quite easily along the membrane. Why potassium cannot go through plasma membrane so easily? Do you know the explanation why potassium cannot go through without any support? Siamin, do you know?
Sorry, I don't know.
You don't know. Okay, someone else, maybe Mariam. Do you know why potassium cannot go through? Look at the charge. It has a charge. It has a positive charge. Do you remember what is the quality of phospholipids? So this is phospholipid. Yeah, you know, this is fatty acids, and this is the charged zwitterionic part. So it has a positive charge as well as the negative, positive cross, and the negative one. So, no, any idea, Marek?
Well, if it's with the charges, doesn't it get repelled without the shield?
There is a repulsion of, there are two interactions. It's a repulsion between the positive charges and also attraction with the negative one. So first, the potassium will be repulsed because first goes the positive one and secondly goes the negative one. So in the first barrier is the repulsion with the positive charge, and then is the trapping by the negative charge, which is situated on the orthophosphoric acid part of phospholipids. So that's the reason. That's quite easy. The explanation is quite easy. So we need something which isolate the potassium charge, and by this way it can smoothly go across the plasma membrane. Is it clear? Sunny, Omar, Mariam?
Yes.
Okay.
Yes.
Great. So in every question, just think about it. Usually the explanation, it's quite simple. You can just see the quality of the particles, and you can maybe assess all or majority of explanations from the quality of the particles, or especially it's valid in chemistry. Just think about it. Do not memorize. You have to memorize a little bit, but do not memorize everything. Try to understand to the problem, first of all. Okay, this is grametidine A. This is the lipophilic, so you will see. So chemically, what is that? Just look at this one. So, do you know what's that? It's oligoprotein. So you need, it's a sequence of glycine, alanine, leucine, alanine. So this is the abbreviation for amino acids. So, and if you have a sequence of amino acids, we are talking about a polypeptide. So this polypeptide has the spiral conformation, alpha helix, and it's immersed in 1/2 of the plasma membrane. The constitution of grametidine A, it's from two parts. One part is immersed in the outer part of the membrane, and the second part is immersed in inner part of the membrane. If they are separated, this channel is closed. Yeah. Once they became in contact, so this is closed. This is closed. Once they became in contact, they create like a tube together, and by this way, the ions may go through smoothly. It's again the isolation of their charge. Yeah, is it clear, Omar? Is it clear? Siamin, is it clear?
Sorry, can you once again?
Yeah, sure. I know why I'm asking. So grametidine A, it's a polyprotein, polypeptide. Yeah, polypeptide maybe it's better formulation. So it's a sequence of amino acids. It creates an alpha helix structure, as you can see in this picture, like a spiral structure. It has two parts. The first one is immersed in outer part of the plasma membrane, outer part, and the second one is immersed in inner part of plasma membrane. Simply with the shift of these two parts, we can have the channel closed, it's this point, channel closed, or open. They just shift together and they are coordinated in one line. By the coordination into one line, they are created a tube, and through this tube, the ions, in cations in this case, smoothly goes through. Is it clear now?
May I have a question?
Yes.
What causes the shift in the two layers?
Yeah, that's a clever question. I expected that question. There are two points. Two ways how to change the conformation of these two parts. First one is by the attachment of some chemicals, so by sensor. Yeah, they change the configuration by some messenger. This is the first one. And the second point is by the change of the charge on the membrane, so by voltage. Voltage change its structure, or chemicals change its structure. Yeah, two main ways how to change the structure. Is it clear? I changed the voltage of whole plasma membrane, and by this way I changed the conformation. This is the first way. And the second way, I have something, some chemical point, which can be attached by, for example, to this side, and it really yields to the shift of this compound to equalize and creation of the tube. Is it clear?
Yeah, thank you.
Welcome. Okay, what we can say about the channels in general. So first of all, we can say that the channels which are created in plasma membrane, they allow the passage of some compounds through the membrane. Some of them are open all the time, but some of them are closed, like we saw in this case. Yeah? And we can control if they are opening by voltage, as I mentioned before, by change of the charge, and also by some ligand, by some messenger. Yeah? You know, the activation can be through, for example, acetylcholine. You know how do you know probably you know the principle of the transfer of the signal from the brain via motor neurons to the muscle. So the messenger in this point is acetylcholine. So, for example, acetylcholine. So E is missing, acetylcholine. Okay. And the second way is via the change of the voltage. Also, we can find in our tissues the interconnection between the cells. Usually it's in tissues because the same quality cells are the tendency to just split together with the gap junction. So this gap junction has the advantage because the interconnection between two cells is pretty fast. They are created like some channels or bypasses. So, however, it's usually just in one range of the tissues, like in liver tissues, or especially in one type of the cells, like beta cells within pancreas cells and so on. Langerhans isolates, they have also the gap junctions. So this is what can be said by channels in general. And now we are getting to the active transport. So active transport usually goes against the concentration gradient. So I have a room which is crowded full of people and I'm pushing more people inside, and because of I'm pushing them, they really wouldn't to go there because it's very messy, it's very little space there, so I need some energy to push them there. So I have two sources of energy. The first one is ATP and the second one is electrical gradient. I would like to show you this little summary, and then after this summary I will show you a video. So first of all, we mentioned the passive transport, which can go just with simple diffusion, using channels and using some carriers. So this passive diffusion, it goes along the concentration gradient, yeah? From the concentration, from the higher concentration to the lower concentration. On the other hand, the active transport requires some energy, therefore we call it active, and it can go from the less concentrated part to the more concentrated part. So we are pushing people to the crowded room. So I will show you the video. Give me a second. I have to stop recording for this moment.